Ladies and gentlemen, I'm Megasit Somsuk from Mahidol University. It's my honor to introduce our plenary speaker this morning, Professor Drew Weissman. He's a distinguished physician scientist at the University of Pennsylvania. He's globally renowned for his groundbreaking work in mRNA technology, which laid the foundation for the development of mRNA-based COVID-19 vaccine that we use worldwide. His seminal research, particularly in collaboration with Professor Kathleen Calico, led to the discovery of method to modify synthetic mRNA to make it more stable and less inflammatory, enabling its use as a safe and effective platform for vaccine. This technology became the scientific basis for the Pfizer, BioNTech, and Moderna vaccine that playing a critical role in combating the COVID-19 pandemic. For his pioneering contribution to medicine and public health, Professor Weissman has received numerous prestigious accolades, including the 2023 Nobel Prize in Physiology or Medicine. His work exemplifies the power of translational research in addressing urgent global health challenges. Without further ado, please join me to welcome Professor Drew Weissman.
Thank you very much. So, as you can imagine, I'm invited to attend and talk at a lot of different places all over the world. The one thing that is always at the top of my list that I try never to say no to is when I talk to students, because I find that that's the most important. And don't tell your PIs this, but they ask much better questions. So I look forward to having some great questions at the end of my talk. What I wanted to talk to you about is a broad overview of nucleoside-modified mRNA. Hopefully, everybody in this room has had at least one immunization with that vaccine. But what you don't realize is that the potential for other uses, other things that can be done, is enormous. There are hundreds, 500s, of clinical trials going on right now that include things like gene therapy, in vivo CAR T therapy, cancer therapies. The amount of potential therapeutic applications for RNA LNPs is enormous. So I'll go through a few of these. I always like to start when I'm talking to students because this is something that the directors of this course always ask. And they want me to tell you what it takes to be a successful researcher. I have no idea. I can tell you what made me successful, but that certainly isn't going to apply to most other people. There is no set personality, set of beliefs, set of interests, approaches that makes somebody a researcher. There are a few basic qualities that I always find to be incredibly important for somebody to think about and become successful in research. The first one of these is curiosity, creativity. Because researchers spend most of their time thinking of new ideas, new hypotheses, new approaches. Most of them, 90+% of them, never work. But you have to keep trying. And it's important to think of anything you want to research and then look at it and decide, is this worth spending time discovering? And if good researchers say yes much more than they say no, because it's the curiosity, the wonderment about how things work, how things function, that makes researchers successful. When you're playing research, it's a lot like playing the old whack-a-mole game that I'm sure many of us did when we were young kids, where the moles pop out of the ground and you have to hit them. It's the same thing in research. There are hundreds or thousands or more hypotheses, ideas that you want to approach, and you're constantly hitting them, trying to find the right one. I was in a Gothic church in Spain a couple years ago, and there was a painting on the wall of the church. And what I learned from this painting is that in the old days, it was the other way around. The mole hit the people who were popping out of holes. I'm not sure which version I prefer, but that's how it started. So I'd like to start by talking about the timeline of mRNA. And I do this because when the pandemic first hit and when the vaccines were first released, I spent a lot of time, most evenings and weekends, talking to lay audiences. And the purpose was to first explain to them what COVID-19 was, and then to explain to them what the RNA vaccine was, and why it was important for them to take it, how it worked, why it was safe. And one thing that I would always get a comment on from somebody in the audience was, oh, I don't know if I want to take it. It was invented in 10 months. And I have to be polite, and my response would be, well, actually mRNA was discovered in 1961. That was around the time that I was born, a long time ago. The first time RNA was given to humans, given to patients in a clinical trial was the late 1990s. That was probably well before all of you were born. So RNA has been around for a long time. It's been in people for a long time. It's not a brand new technology that was invented in a matter of months in the setting of a COVID emergency. I came out of the NIH where I did my fellowship, and I worked for Tony Fauci. You probably know who he is. After I left NIH and started my own lab, I still went to HIV meetings, and I would run into Tony and to Gary Nabel and to many other luminaries in the HIV field, and they would ask me what I was up to, what I was doing. And I would sit down and tell them about the RNA that Katie and I were working on. And they would listen, they would nod, they would smile, and often at the end they would say, you know, Drew, you really should do something more worthwhile. RNA is never going to work, it's never going to be useful, it's never going to be a good therapeutic. And I would try and argue with them a little, and I would point out that protein therapeutics is one of the most advancing fields in pharmaceutical science and pharmaceutical companies. This is what it takes to make a therapeutic protein. You start with a 50,000 liter drum of CHO cells or other cells. You collect the supernatant from those cells. You then have to develop a purification approach in order to purify that protein away from all the cell culture contaminants. If you want to switch to a different protein or different antibody, you start over. everything starts over. This is very expensive. In the United States, the cost of monoclonal antibody therapy is $50,000 to $200,000 a year. And what Katie and I believed when we first started this was that mRNA could replace protein delivery. And instead of relying on a tumor cell line in a 50,000-liter drum, we would use host cells as the factory to make the protein. And we would deliver the mRNA that encoded the protein of interest. And the advantages of this were that each mRNA makes thousands, if not tens of thousands of proteins, so a huge amplification. Host cells are making the protein, not a tumor cell line in culture. So host cells will fold it correctly, glycosylate it correctly. So it's much simpler because you don't need host cells. It's just an enzymatic reaction. So this is what I would argue to them. Needless to say, I didn't get very far. When you now think about moving into the vaccine field, in general, live virus vaccines are the most effective. The first vaccine developed, cowpox, was a live virus. Many of the vaccines, MMR, influenza for young kids, are live viruses. They work because they infect our cells and they produce protein for a long time. Unlike a tetanus vaccine where the protein washes through your body in a few hours, the immune system likes to see extended durations of protein production. The problem is that the FDA and regulatory agencies hate live virus vaccines. They can change, they can become more pathogenic, they're expensive and difficult to make. RNA is a single enzymatic reaction, very simple. This is the broad spectrum of different types of vaccines. So you can see the live attenuated viruses, the killed viruses, subunit proteins, DNA, RNA, and viral vector are the newest approaches. This is the data that we generated in our first experiments trying to use mRNA as a vaccine. You don't have to know what this is. It's measuring neutralization of influenza virus. But what you need to pay attention to is the scale. This is an activated virus. This is live virus in the nose. This is mRNA encoding influenza hemagglutinin. It's 50 times better than an inactivated virus. When you talk to a pharmaceutical company or a biotech company, they're interested in vaccines that are better. Twofold better is wonderful to them. Fourfold, they go crazy. 50-fold caught their attention. The way vaccines work is they activate the acquired immune system. And there's two broad avenues, sections of the acquired immune system. There are T cells. T cells recognize foreign antigen expressed by host cells, or there's antibodies. And B cells make antibodies that can kill at a distance. There's supposed to be words here. I'm not sure I can mimic them, but we'll give them a chance to. This is a coronavirus attacking antibodies and then activating the immune system. So a patient just got a vaccine, and the vaccine induces antibodies that recognize the spike protein. And these antibodies look for coronaviruses in the respiratory tract or anywhere else in the body. And by binding to the surface spike, neutralize their ability to infect other cells and prevents their infection. They can also be bound by other immune cells that can kill the virus. These are killer T cells. The killer T cell is in green, and you can see it goes around searching, in this case for cancer cells, and it engulfs, it attacks, and it delivers toxic proteins and kills the virally infected cells or the tumor cells. They can also repeatedly kill, which means one cell can kill many different tumor cells, and they go around the body searching for cells that have the antigens that they recognize. Once they find them, they form synapses and deliver toxic proteins that then kill the tumor or virally infected cell. So now what I want to talk to you about are some of the many different things that can be done with RNA LNPs. I always put this slide up, mostly because it upsets pharmaceutical executives, because they believe it oversimplifies making an RNA vaccine. But in the lab, this is how easy it is. You only need a sequence. So in late January, when the Chula researchers released the sequence of COVID-19, we and others looked for the spike protein, took the sequence, and made a vaccine in hours. Once you have the sequence, you make a plasmid, a piece of DNA, that you produce RNA off of in a simple reaction. You purify the RNA, you mix it with lipids, they self-assemble into a lipid nanoparticle, you filter away the salts and ethanol, and your vaccine is done. We can make vaccines in a few hours. So I wanted to talk to you about a couple different vaccines for different members of the pathogen families. And the first thing to realize is that pathogens have a wide variety of sizes that associate with how they function, how they infect cells, how they infect organisms, how they work. But the range is quite wide, from 0.05 microns to hundreds of microns. Viruses are the easiest pathogen to make a vaccine against. The reason for that is viruses require a host cell to infect, and it uses host cell machinery to replicate. That infectious step requires binding to a cell surface receptor to allow the virus to enter the cell. So if you make an antibody that binds that cell surface protein, it neutralizes the virus. It prevents it from infecting the cell and stops the infection. This is the classic figure from the phase 3 clinical trial of the Pfizer COVID-19 vaccine. And what you're looking at here, this is a measure of incidence of disease. The blue are unimmunized people. So in this population, COVID-19 increased at this rate. These are the people that were vaccinated. And you can see within 10 days of the second dose, the line flattens out. People stopped getting infected. The vaccine was 95% effective at blocking infection. Now, this, of course, only worked for the very first COVID-19 virus before variants and mutations started to show up. We've also used mRNA to make vaccines against bacteria. Bacteria usually, some do, but most don't infect cells. They cause disease by infiltrating tissues, releasing toxins, releasing other factors that then injure the tissue and cause the disease. I'm going to show you data from a vaccine against a bacterium known as C. difficile. C. difficile is a resident of our GI tract. In many people, it's part of our microbiome. If people receive antibiotics, it upsets the microbiome and can allow C. difficile to expand and cause disease. Unfortunately, over the past couple decades, it's also become a community-acquired infection that can occur in situ in people outside of a hospital. This gives you an idea of the burden that C. difficile leads to, multiple billions of dollars a year, because it attacks in many ways. It causes a very severe disease that people can die of, but it also results in extended hospitalizations, because the problem is we don't have a good way to treat C. difficile. There are a variety of antibiotics that are used. 30% of treated people recur and have multiple infections. We've tried things like stool transplants that have some more efficacy. But in the absence of an effective vaccine, it's a severe disease that's very difficult to treat. The main factor of C. difficile disease are toxins that are released by the bacteria. The bacteria don't cross our GI tract. They damage it, but they don't cross and infiltrate tissue. What they do is they release toxins that are taken up by host cells that enter our circulation, impair the immune system, impair a variety of things. So what we've done here is we've made two vaccines. One of them includes both toxins, A and B. That's the bivalent vaccine. The other, we've added a third antigen from the bacteria, and this is a cell surface secreted protein. The importance of what you can see here is these are animals that were vaccinated, and we challenged them with C. diff. So they had very high levels of toxin for 3 days. And then over the next week, they reduced. But the green, this is toxin-only vaccine, never got rid of the toxin. That means that the bacteria remained in the GI tract, which allows it to return at some point in the future. The animals that received the trivalent vaccine, they cleared toxin from their GI tract. They also cleared the toxin from, they cleared the bacteria from their GI tract. That's called decolonization. To us, this is an effective vaccine. It treats the acute disease, but it also gets rid of the bacteria and prevents it from colonizing and ever causing disease again. This is another advantage of RNA vaccines. You can add many different RNAs into a single vaccine. We have vaccines that have 20 RNAs in them for 20 different antigens. In general, most protein or live virus vaccines are limited to typically 3 different antigens due to competition between the antigens. RNA doesn't suffer that. So we're using RNA for a large number of additional things. These are some of the vaccines we're working on. As you can see, they include very difficult, if not impossible to treat diseases: HIV, HCV, herpes virus, malaria, CMV, TB, and many others. And we're seeing effectiveness in a lot of these early clinical trials. But we've gone further. We're now using RNA to make vaccines for food allergies, peanut allergies. We're using RNA to treat environmental allergies. We're using it to treat autoimmune diseases. Autoimmune diseases in general are treated with immunosuppressive drugs that reduce the symptoms but don't ameliorate the disease. We're hoping that with RNA vaccines that work in an antigen-specific manner, we'll be able to better cure the autoimmune disease without immunosuppressing the patient. And finally, we have a number of cancer vaccines in clinical trials. Some of them have already been successful. A couple months ago, groups from New York released a study where people with pancreatic cancer had disease remissions, disease clearances with a cancer RNA vaccine. If you know pancreatic cancer, nothing works for pancreatic cancer. If you don't catch it early, it's a terminal disease. But this vaccine, 25% of the people are still alive. There's cancer vaccines for melanoma have also shown similar efficacy. Now I want to move on to mRNA therapeutics. So in addition to being a vaccine, to triggering the immune system to respond to a foreign protein, you can also deliver host proteins or therapeutic proteins with mRNA. You can deliver monoclonal antibodies, you can deliver replacement proteins, you can deliver proteins that alter functions or improve functions. It's in clinical trials to treat cystic fibrosis, and a variety of liver genetic diseases. But there's a problem. Lipid nanoparticles, which are the accepted way of delivering RNA, target the liver. They bind ApoE, a serum protein, and go to the liver. So we were presented with a problem over 10 years ago. How do you deliver RNA to other cells, tissues, and organs? We started off by looking at three simple approaches, targeting endothelial cells, T cells, and stem cells. And we took a very simple approach. We attached targeting moieties. Here, they're antibodies, but they can be ligands for receptors. They can be peptides that bind surface proteins. They can be DNA aptamers. They can be sugars. They can be anything you want. But we attach those to the surface of the lipid nanoparticle. Our interest is to use this to treat initially sickle cell anemia. Sickle cell anemia affects, it's actually 350,000 people a year, mainly in sub-Saharan Africa. The issue, though, is that last December, 2 new clinical trials were approved to treat sickle cell anemia. What they do is they take bone marrow out of the patient. They put it into culture in a specialized hospital lab. They infect it with a lentivirus that alters the genome. They give the patient chemotherapy, and then they give them the cells back. The issue is that it's predicted to cost over $3 million a person. So if you multiply 3 million by 300,000, you've bankrupted the world. You also don't have those laboratory facilities available in low and middle income countries like sub-Saharan Africa. Our idea was we could use mRNA LNPs to target the cells of interest. For sickle cell, it's bone marrow stem cells. They produce all of the blood cells and a few other cell types in our body. By targeting a lipid nanoparticle with a targeting moiety, we could deliver them to the bone marrow stem cells that produce all of our blood cells, including red blood cells. In the RNA, we could encode a gene editing enzyme. In this case, we use what's known as a base editor. It uses a guide RNA to change bases at specific sites. There are 2 mutations that can be targeted with sickle cell. One is in the beta hemoglobin. The other is a locus known as BC11A that drives fetal hemoglobin. By using a base editor for either of those and targeting bone marrow stem cells, we believe we can treat sickle cell anemia with a simple injection of RNA LNPs. So here we're using a mouse where if we deliver a gene editing protein, the cells that are targeted change color. It makes them easy to follow. These are mice that were treated once with a bone marrow targeting LNP and a gene editing enzyme. And as you can see, the red are the treated group. A single treatment led to nearly all of the red blood cells being gene edited with a single injection into this mouse. My dream is to someday get an igloo cooler, a frozen cooler full of RNA LNPs, go to anywhere in sub-Saharan Africa or India or anywhere in the world, and set up a table, put my cooler down, and line people up that have sickle cell anemia and give them an injection and cure their disease, one after the other. We can do thousands of people, if not tens of thousands of people a day. We've looked whether this works. So we used a bone marrow stem cell targeted LNP, and we mixed them with bone marrow stem cells. These stem cells are from a patient with sickle cell. So you can see these arrows right above them are sickled red blood cells, and there's many of them in this patient. Here, they've been treated with the LNP targeted to stem cells, and there's no more sickle cells. Here, we just quantitated them. Nearly all of the sickle cells disappeared, and the amount of correct hemoglobin greatly increased. So we cured bone marrow stem cells in culture. We're now doing this in mice and getting similar results. We hope to move to people within the next year. So my views of future potential therapies at this early stage are you can probably do just about anything. You can edit genomes, you can deliver therapeutic proteins. The problem is the delivery. For bone marrow stem cell targeting, there are also many different things you can do. I talked to you about gene therapy, but you can use RNA LNPs to alter function of bone marrow cells. Some people don't make enough red cells or granulocytes or platelets. We can give them therapeutic proteins in the bone marrow with no adverse events that will improve that function. A type of therapy that's often used on difficult to treat cancers is known as bone marrow transplants. And that's where you take bone marrow from a healthy individual. Identical twin is always the best, but most of us don't have them. And you transplant them into patients. The difficulty is that you have to kill off all of the host bone marrow stem cells. That's done with high-dose chemotherapy. And there's a toxicity to that and a mortality to that. We're now using RNA LNPs targeted to stem cells to selectively kill them off with no adverse events. So there's many different things that can be done. I wanted to talk to you about a new approach to cancer therapy. So CAR T cells were originally developed in Israel, but Carl June at Penn clinically developed them and clinically tested them. What a CAR T cell does is it mimics a cytotoxic T cell. I showed you the video earlier of how they go around and kill tumor cells and virally infected cells that they recognize as foreign. The way a CAR T cell does this is it takes the T cell receptor. The T cell receptor recognizes MHC peptide, so they only work in the person that generates them. A CAR T cell exchanges that exterior portion with a piece of antibody that recognizes a tumor-associated antigen. The first CAR T cells used a CD19 antibody. CD19 is expressed by early and late B cells, but it's also expressed by many different kinds of B cell leukemias and lymphomas. This is what it takes to make a CAR T cell. They're made from each individual patient has their own CAR T cells made. You start by leukapheresis. That's a big machine that takes blood out, spins it around, takes out the white cells, gives you everything back, does that over and over for a few hours. You then take those 2 billion white cells and you infect them with a lentivirus. The lentivirus delivers the CAR gene, the gene that makes the CAR molecule. You then have to expand those cells, typically for about 10 days, to make enough cells to treat a patient. You then have to give the patient chemotherapy, essentially to make room. You have to kill off lymphocytes to allow these lymphocytes to go in, set up shop, and divide. It takes 2 weeks for the B cells to be affected because that's how long it takes for the CAR T cells to set up shop. And during that time, they have many different adverse events. Our idea was we can make CAR T cells in vivo, in the body. And the way we would do that is we take LNPs and add targeting molecules on them that targets T cells. We would encode the CAR molecule in the mRNA. So the LNPs are injected, they find T cells, they deliver the CAR messenger RNA. The T cell puts the CARs on their surface. In this study, we used a CAR that recognized activated fibroblasts, and we used a model of cardiac fibrosis. So if you make mice or people hypertensive, they fibrose their heart. This CAR kills the cells that induce the fibrosis. These are measurements from an echocardiogram. What it's important to see is that the gray is untreated animals. The orange are hypertensive, fibrotic animals. And what you can see is that the diameter of their ventricles increases and the amount of blood they can pump out in the bottom is decreased. That's because the heart is stiff, it's fibrotic. It isn't moving very well. In red, these are animals that we treated once with LNPs that targeted the T cells and delivered a CAR T. And you can see their heart function returns to normal. So a single treatment with an off-the-shelf RNA LNP returned their heart function to normal. We took out their hearts and stained them for fibrosis. That shows up in red. And you can see in untreated animals, there's very little red. In animals that are hypertensive and fibrotic, quite dense red staining. On your right, a single treatment with LNPs gets rid of the fibrosis. We've now taken this into humans. CAR T cells in general, when they're made with a lentivirus, are long-lived. The lentivirus inserts into the genome, and as long as the cells are alive, the CAR Ts are available. That's great for cancer. It's not so good for cardiac fibrosis because we have fibrosis in many of our tissues that keeps our body structure intact. So the advantage here of RNA is it's transient. You only have activity for a few days. What we wanted to try was using in vivo CAR Ts to reset the B cell system. That's important for a number of diseases, particularly autoimmune diseases, where we know abnormal B cells make antibodies and induce responses against autoimmune antigens. And the idea is that if you can deplete those diseased B cells and replace them with naive B cells, you can treat the disease. So these are lymphocytes from rhesus macaques that we treated with our RNA LNPs using a targeting antibody that works on macaques as well as humans. And you can see that a single treatment of cells at moderate low doses gives you 80% of the cells expressing CARs. We treated macaques in vivo. We didn't know how to treat them because we had never done this in a macaque before. So we treated them every 3 days. We gave them a dose for 2 doses and then looked at CAR expression. And you can see by the 3rd dose, nearly 80% of the T cells in vivo have CARs on them. We then treated macaques. We gave them the LNPs in vivo. And in the top, we're simply measuring B cells. So PBS-treated animals, their B cells just bounce around a lot. But starting at the lowest dose, within 6 hours, all of the B cells are gone. And as we increase the dose, the duration of their disappearance increases. But more importantly, when we look at the tissues, this is a lymph node, but we've looked at every tissue with lymphoid material. As we increase the dose of CAR Ts, we deplete the B cells from the tissue. So essentially what we're doing is we're depleting just about every B cell in these animals. When we do that and we follow what happens over time, what we see is that after 3 to 5 weeks, depending on the dose, the B cells come back. But what come back are naive B cells. None of the memory cells remain. They're gone. The naive B cells are naive. They don't recognize any antigens, and they take over the immune system. A few patients have been treated with CAR Ts for 3 years. They have not recurred their disease. So we think that this could have long-term efficacy. But more important, if the disease comes back, you treat them again. You give them another dose of RNA LNPs. Very simple. These in vivo CAR Ts can be used for other diseases in addition to autoimmune diseases. Now, when you do CAR T therapy, there's a mortality rate of 2 to 5%, depending on the site. So that means CAR Ts are generally done for cancers that are incurable or autoimmune diseases that have high mortality rates. You couldn't do it for an autoimmune disease where people don't die, things like type 1 diabetes or thyroiditis or rheumatoid arthritis, where the autoimmune disease doesn't shorten lifespan. In vivo CAR Ts have no adverse events. So we're now looking at using these for all of those other autoimmune diseases. It can be used to treat other chronic diseases, things like HIV or fibrotic diseases. There are many fibrotic diseases. There's interstitial lung fibrosis, where currently there's no cure. Most of the muscular dystrophies result in extensive fibrosis, and the symptoms are from the fibrosis before they're from loss of muscle, including cardiac muscle. So there's a variety of diseases that can be effectively treated and treated all over the world. I always throw in CNS targeting. For this audience, you guys are young enough where you don't worry about this yet. If you look at it, the handful of older people, it's something we're concerned about. When I was a resident many decades ago, this was our nightmare admission on Friday nights. It was a guarantee. The stroke patient. And this is a CAT scan, and you can see right here, this is dead brain from a stroke. The patient likely walked in, they were awake, they might have had some difficulty talking, but they were otherwise healthy, taking care of themselves. The problem is what happened over the next week, and that's seen in this CAT scan. The stroke has expanded to nearly the entire half of the brain. These patients didn't walk out of the hospital. The reason that occurs is dead brain releases inflammatory mediators, and that inflammation expands the stroke. So we wanted to know, could we use RNA LNPs targeted to the brain to block that enhanced inflammation and treat stroke? So our collaborators have a machine that allows them to inject inflammatory modulators into one half of the brain. And we did that. We injected tumor necrosis factor alpha. As you can tell from the name, it's an inflammatory mediator. We only injected it onto one side. And then we treated the animals with LNPs that were targeted to the brain, and the RNA encoded an anti-inflammatory molecule. And what you can see is that in black, these are saline-treated animals, and we're measuring leakage of the blood vessels into the brain. That causes increased pressure and brain death. And there's very high levels of it on the side where the TNF was injected. In red, that's the LNP that was targeted to the brain and contained an anti-inflammatory mediator. And we can see the level of vascular leakage was unchanged from the untreated side. This is now going into clinical trials as an acute treatment for a variety of different brain diseases and strokes. So the potential for mRNA therapies is enormous. I joke to my young people, we haven't thought of most of the things you could probably do with RNA, and that's their job to figure out. You can do a variety of in vivo modulations of cellular function. You can gene edit stem cells. Those stem cells are present everywhere in the body. You might have heard for a couple weeks ago, a 6-month-old had an ammonia genetic disorder. The child never left the hospital, and these children don't. They're usually treated with diet. You don't give them any protein to reduce the amount of ammonia that they make. They die very young ages with no treatments. This child They sequenced his genome and found the defect in the enzyme that made high levels of ammonia. And they made a guide RNA LNP treatment, treated the child, and within a couple weeks, he was eating anything he wanted and never had an elevated ammonia level. It's been almost 6 months. He's home, playing with his friends, eating whatever he wants. His disease was cured with a simple RNA LNP treatment. That can be extended to the thousands of other genetic diseases in any organ that we can deliver RNA LNPs to. So I wanted to mention, since you guys are young and you're now the future of RNA LNPs, I wanted to talk to you a bit about vaccine misinformation and disinformation. You probably don't realize
Anti-vaccine claims, many of which come from a handful of influential social media figures. NPR's Shannon Bond reports.
This problem is nothing new to people who track misinformation. Many misleading claims and outright lies about COVID vaccines mirror what's been said about other vaccines. John Gregory is deputy health editor at NewsGuard.
It's almost like conspiracy theory Mad Libs. They just inserted the new claims.
And it's not just the claims that are familiar, it's who is making them. Just 12 people, dubbed the Disinformation Dozen, are responsible for the bulk of anti-vaccine content on Facebook, Instagram, and Twitter, researchers found. Imran Ahmed is CEO of the Center for Countering Digital Hate, which identified these figures.
The Disinformation Dozen produced 65% of the shares of anti-vaccine misinformation on social media platforms. They're producing the most shared content.
Some focus on natural health, some sell supplements and books, and many have long spread scientifically disproven medical claims and conspiracies. And they're still doing it now, proving how hard it is to stop hoaxes on social media. Ahmed says the claims they make about COVID come straight from the anti-vaccination playbook.
Denying that COVID exists, claiming that false cures are in fact the way to solve COVID and not vaccination, decrying vaccines and decrying doctors as being in some way venal or motivated by other factors when they recommend vaccines.
Lawmakers and state regulators have urged Facebook and Twitter to ban all 12 accounts. A note, Facebook is among NPR's financial supporters. The companies have stepped up the fight. They've labeled posts, even removed falsehoods, and banned people who repeatedly shared debunked claims. Still, the Disinformation Dozen are easy to find on social media. Ahmed says sometimes they skirt the platform's rules by using codes.
So, for example, instead of saying vaccine, they may, in a video, hold up the V sign with their fingers and say, if you're around someone who has been hold up V sign, X might happen to you.
Or they take something true and distort it, like falsely linking a famous person's death to the fact they got a vaccine days or weeks earlier. Facebook says it now limits the reach of posts that could discourage people from getting vaccinated, even if they don't explicitly break its rules. Some of the Disinformation Dozen have toned down their posts, but the cat and mouse game continues. Take anti-vaccine activist Robert F. Kennedy Jr.
I have to post like unicorns and kitty cat pictures on there. I can't post anything. It doesn't matter how true it is.
Facebook, which owns Instagram, kicked Kennedy off the photo sharing app in February, but he's still on Facebook. There, he advertises his website and newsletter, where he makes claims that would break Facebook's rules if posted there. Kennedy told NPR he's never posted misinformation and accused Facebook of censorship. He says the crackdown has sapped his ability to raise donations.
It's cost us hundreds of thousands of dollars.
Even as the social media companies have gotten tougher recently, researchers worry the persistence of these hoaxes will further erode confidence among people who are already hesitant about the COVID vaccines. That's especially concerning as shots roll out for kids 12 and up. Jessica Calarco is a sociologist at Indiana University. She's found more than a quarter of parents don't plan to vaccinate their kids.
So many of these moms are turning to Facebook, are turning to Twitter, are turning to other social media platforms. And they're saying, every time I open my phone, I see something different.
Like stories about bad side effects. Even some parents who have been giving their kids routine childhood vaccines said they're unsure about COVID jabs.
And those kinds of social media-based reports are, in many mothers' minds, kind of weighed equally against the kinds of expert medical recommendations coming out of things like the CDC.
And that could make it hard to push down infection rates. Shannon Bond, NPR News.
So I just wanted to highlight
In the fight to persuade skeptics to get COVID-19.
One element that RFK Jr. has highlighted through his entire career and made all of his money off of, which is the link between the MMR vaccine and autism. What you don't know is that that came out of a single study of a little over a dozen people by a pediatrician named Wakefield that was found to be an absolute lie, was found to be medical fraud. He actually went to jail because it was found out that he did it because he was working on an alternative vaccine platform to MMR. And he published this paper simply to make MMR bad, to make people say, oh, I don't want to take that vaccine. It causes autism. There have been hundreds of studies and hundreds of thousands of people over the past 40 plus years that have shown no link between autism and MMR or any vaccines. But it's still the highlight of what our anti-vaccine people say. To me, the big problem is summed up in a quote by Mark Twain, who said, "A lie can travel halfway around the world while the truth is putting on its shoes." People love conspiracy theories, love good lies. They have little to no interest in the truth. Anti-vaccines have been around as long as there were vaccines. So Edward Jenner in 1796 developed a smallpox vaccine. He noticed that milkmaids milking cows weren't getting smallpox. And when he looked at the cows, they had a similar looking pox lesion. So he took the smallpox and he used that to immunize people. Right after that, paintings like this appeared. And here you can see, here's Jenner regally giving vaccines to people. And you have cows coming out of people's arms, out of their mouths, people turning into cows. That's how the anti-vaxxers described their dislike of vaccines back then. So anti-vaxxers aren't a new thing. They've been around forever. What's new is social media that allows anybody to talk to thousands to millions of people. The last thing I wanted to talk to you is something that I've worked on since I started research: vaccine and therapeutic efficacy. This came to light for the world during the COVID pandemic. And people started to look when the RNA vaccines were first released. Within months, the entire populations of the United States, Western Europe, and a handful of other wealthy countries were completely immunized. Everybody in the country received 2 immunizations within months. But low and middle income countries in sub-Saharan Africa, Southeast Asia, South America, it took them over a year before they received their first RNA vaccines. and they never received the same number that the US and Europe did. This is the population of the world. And as you can see, the majority of people are in sub-Saharan Africa and Southeast Asia. This is where the money is in the world. The money is principally in North America, Western Europe, and a few other scattered countries. This is where malaria is. Similar for TB, for sickle cell, for HIV, for numerous other diseases. They're where the people are, not where the money is. That causes a problem for pharmaceutical companies. Pharmaceutical companies spend hundreds of millions of dollars developing new vaccines and therapeutics. So they do it in a way that they know they'll get their money back. Pharmaceutical companies have no interest in making a malaria vaccine, or making a TB vaccine, or making new dengue vaccines or Zika vaccines. The list goes on, because they'll never make their money back. That's why we believe in developing equity. The first place I did this is at Chulalongkorn University in Bangkok with Kiat, who was the lead of their VRC. We started working together 10 years ago, where they became interested in RNA vaccines. And I brought them to my lab, and I visited their labs, and we established mRNA LNP research in Thailand. When the pandemic hit, it was probably around March of 2020. I was talking with Kiat on our monthly calls, and he was lamenting that the health ministry, the government of Thailand, was concerned that any vaccines made in the West would take years before Southeast Asia, Sub-Saharan Africa, South America ever received them. And he was right. But he talked with the royalty, with the government, with donors, and they gave him money to make his own RNA COVID vaccine. So we started working together, and we developed our own RNA LNP vaccine soon after Moderna and Pfizer did. This is the phase 1 clinical trial of that vaccine. Now, I included a blue-green bar here. That's the Pfizer vaccine. As you can see, the vaccine that Kiat and I made was better than the Pfizer vaccine. It gave higher levels of neutralization than the Pfizer vaccine. I don't tell that to Pfizer, but Kiat is excited by this. We had a problem. We didn't have a place to make the vaccine. So whenever you make drugs or vaccines, it's called GMP, Good Manufacturing Practice manufacturing. It's a highly specialized way of making drugs and vaccines. It's negative pressure, multiple people, specialized environments, specialized equipment. All of the components of everything used are made under similar conditions. It's very expensive to make and very expensive to maintain. Thailand didn't have a GMP site to make RNA vaccines. So we helped build one. And we have a GMP production site right outside of Bangkok that's making RNA LNP vaccines. Kiat now has 10 vaccines that he's developed, his group has developed in clinical trials. Things for diseases like dengue, tularemia, dust mites, Zika, and numerous other diseases. The hope is that they'll be able to develop these vaccines and produce them locally and distribute them locally to the regions that have these diseases and need them. Soon afterwards, I started working with the WHO, and we built 15 more GMP sites in low and middle income countries around the world. These were supported in part by the WHO and in part by the countries. They are spread across low and middle income countries around the world, in Caribbean, South America, Sub-Saharan Africa, Southeast Asia, Eastern Europe. We actually have a GMP site that we built in the Ukraine during the war. How they did that, I can't imagine, but it's up and functioning and making RNA. There's no place to do any experiments, but they can make RNA. The idea is that the next step we're working on is we're developing the basic science research, as I did with Kiat 10 years ago. And the ultimate idea is that we'll have RNA researchers in local universities and companies who are developing new RNA therapeutics for diseases that affect their populations. They can do the basic science research. We'll help them set up clinical trials. They have a GMP site to make the drug or vaccine. We'll help them with regulatory activities and with drug distribution. All of these 15 GMPs talk to each other. So the dengue vaccine that Kiat is making will be shipped to South America that has a big dengue problem. The sickle cell anemia gene therapy will be shared with India and with all other countries with significant populations. My ultimate dream, and my wife and kids yell at me when I say this, but I'm hoping someday this can be expanded to other therapeutics, to small molecule drugs, to other types of therapies, and it may become a model for the future. In addition to giving countries, regions access to the most modern therapeutics, it also builds an economy. They'll have reasons to train PhD science students who will have labs to work in, who will have companies making drugs for them. It'll build an economy, and it'll build a new element to these countries' economies. So what I talked to you about today is the enormous potential for RNA, LNP vaccines and therapeutics to treat an enormous number of diseases, and to do so in effective ways throughout the world. I need to thank the people in my lab who actually did all of the work that I described, and acknowledge the rest of the researchers involved with this work that I showed you today. Thank you.
Okay, thank you very much, Professor Weissman. It's time for the question. So if you have any question, please line up at the microphone on this side and on the other side. We have about 19 minutes for question. Okay, first question, please.
Thank you, Professor, very much. We learned a lot about mRNA, LNP, and because of that intervention, you saved lives, millions of lives. Thank you very much. I was looking at your slides, and you talk not only about COVID vaccine, but also about the other treatments for leukemia, etc., etc. And then I was wondering, you skipped, or it didn't mention anything about HIV. So I think the new genes therapy you are talking about, mRNA, LNP, might be helpful for HIV as well. Can you please elaborate a little bit about that and why you skip about that important disease?
So
Thank you very much.
I couldn't possibly talk about everything. And HIV on its own would take a half an hour to talk about. So I didn't put it in. We have two main focuses. We have HIV vaccines already in clinical trials, and we're seeing good results. We believe it's going to be 5 or 6 years, maybe not with our government shutting down HIV research, but we're hoping in 5 or 6 years we'll have an effective vaccine. But we also have cure research. So the problem with HIV is that it integrates into cells that last a lifetime. We're working on ways of using RNA LNP to target those reservoir cells, cut out the HIV genomes, induce immune responses, or deliver CAR T cells or antibodies to develop a cure so people won't have to stay on drugs for the rest of their life.
Okay, thank you. I'll go to the next one.
Hi, thanks for the presentation. I'm Selim Altıntop from Turkey. I actually read your 2005 paper, the original Nobel Prize winner paper, and I resembled that you tested the mRNA versions, in vivo mRNA versions on innate immune system, and the results and conclusions were based upon them. Also, we know that every year 1 billion people, around 1 billion people are infected with influenza, and 400 to 600 of them are dying from influenza every year, and these are mostly children in Africa and developing countries. So I have two questions. The first one is, why haven't you created mRNA vaccines for influenza or similar RNA viruses? Because you know the technology for even more than 20 years, and it is a nice time thinking that you created one for COVID-19 just in 10 months. My first question is that, and the second question is, why didn't you use adapted immune system in your research? Because immune systems differ on every person, every people, and so the vaccine's effects and its responses can also differ. So these are my questions. Thank you.
So we actually started clinical trials for influenza before the COVID-19 pandemic, and they got shelved for a few years. We've been doing trials ever since. What we're working on, we call a universal influenza vaccine. So the problem with flu is that it mutates, similar to COVID, but faster. Every year, you have to make a new flu vaccine. And every so often, you have new pandemics. You have avian influenzas or pig influenzas that cross over. The vaccines we're developing now are more universal. They'll protect against pandemic viruses and mutations longer. So we started with the innate immune system because the entrance of everything into our immune system is via the innate. You have to go into the dendritic cells, the antigen-presenting cells. They then trigger the acquired immune system. So we weren't ignoring the acquired immune system. We were entering the way a vaccine does, through the innate immune system. We've looked, and companies have looked, RNA is very broad. It affects every race. It affects men and women. It affects elderly people much better than protein and other vaccines. It doesn't work well in immunosuppressed people, but that's because their acquired immune systems don't work well. We're looking at ways of addressing that.
But autoimmune diseases are really hard to track because of their unknownity, and also, as you said, RNA viruses have different variants and they continue to create different ones. So how can one universal RNA vaccine could be suitable for all the variants and all the people? Because there are a lot of differentials and different equations. So
Yeah, so our universal vaccines, one of them has 20 different RNAs that have 20 different sources of influenza that gives you the broad coverage. The issue, so respiratory viruses require high levels of antibody to be effective. That means a single vaccine isn't going to work for 10 years for flu. It'll work a lifetime for measles, for mumps, for other, but for respiratory infections, you're going to need a yearly vaccination, even if it's universal. And that's what we're working on. Thank you.
okay, thank you for your answer.
Thank you. Go to next. This one, this side.
Okay. Hello, my name is Poom, and I'm from Thailand. It is a great honor to be given knowledge by such an accomplished researcher such as yourself. So my question is, from your perspectives, what are the major scientific or clinical barriers that still need to be overcome before mRNA vaccines can be widely used for treating or preventing different types of cancer, for example, personalized applications like neoantigen-targeted therapies? Thank you.
So I think what we've learned about cancer, and we've done this ever since we've been studying, is it's likely that a single treatment is not going to be enough. Chemotherapy alone isn't effective in all cancers. Checkpoint inhibitors alone aren't effective. Vaccines alone aren't effective. So I think what we're learning is that any cancer therapeutic needs to be a combination of therapeutics that attack the tumor cells in different manners. So we've got programs that are delivering ASOs to turn off genes, that are delivering Cas9 to turn off oncogenes, that are delivering mRNAs to turn on tumor suppressive genes, vaccines that induce immunity, autoimmune checkpoint inhibitor antibodies. So I think for cancer, it's going to be a combination of therapies. And we have to figure out what's the combination that will work best for every type of cancer.
Thank you very much.
Okay, I'll take that question from that side.
Hello, sir. My question is, can we have vaccine for genetic disorders? And what would be challenges and what are ongoing research?
So to me, the first challenge for genetic diseases is identifying the site that has to be treated. So liver genetic diseases, Krebs cycle, ammonia cycle, that of course is liver. Cystic fibrosis, you have to target the lung and the pancreas. Bone marrow disorders like sickle cell, SCID, you have to target the bone marrow. So the first thing is getting to the tissue of interest. My interest is doing it in vivo because that's the only way it will be applicable to everybody in the world. We can't afford to take bone marrow out of people for $3 million. So we're moving in the right direction. There are multiple reports of liver genetic diseases that have been cured. We're hoping to treat bone marrow disorders soon. There's clinical trials of cystic fibrosis ongoing. We're headed in the right direction, and the data is being generated.
Okay. Thank you, sir.
Okay, next.
Okay, I'm Zhang Lequan from China. Really appreciate for everything you shared today. And my question is that today we mainly talk about the antigen-based vaccines or therapies. And I wonder, is that possible that we can have antibody-based mRNA chain and deliver to the cell, which is maybe more valuable to patients who have immune deficiency, some acute symptoms, or maybe other short-term protections.
No, exactly. And we delivered antibodies with mRNA, I think about 8 years ago. We have a big program in the lab where we're actually delivering mRNA encoding IgA to the nose, into the lungs. And the idea is that elderly are particularly immune suppressed. We can go to nursing homes, and once a week, people get a spray in their nose to make antibodies against flu, RSV, and COVID. We're doing the same for systemic antibodies. Those are ongoing programs. They've been in humans in clinical trials already.
Thank you.
Okay, next one.
First of all, thank you for the interesting presentation. And my question is, how can we accurately deliver mRNA LNP to the intended target site? So are there any strategies beyond physically restricting the injection location?
Yeah, I mean, so we use targeting moieties. They're not perfect. You still get expression in the liver. You still get expression in antigen-presenting cells. If you're delivering a protein that can't be expressed by any other cell, there are ways of blocking its translation using microRNAs or other therapies. It's not going to be, we're never going to be able to have a delivery technology that only delivers to one type of cell. And we just have to figure out how to make it work.
Thank you.
Okay, next one.
Hello, sir. I'm Sim from Myanmar. It's my honor to ask the question. My question is, what are the main barriers of mRNA therapies delivering to the brain, and how researchers are working to overcome them?
Yeah, so the brain, of course, has the main barriers, which is the blood-brain barrier. What we're doing now is we're injecting the LNPs into the brain, into the ventricles, into the cerebral spinal fluid. And that gives us good access to the brain. We still have to figure out how to traverse brain tissue, and those are ongoing. The hope is someday we'll figure out how to cross the blood-brain barrier. So if you're treating a glioblastoma cancer, you don't care if you inject into the brain, because you're going to die of the disease if you don't. If you're treating early or preventing Alzheimer's, you can't inject into people's brains. So you have to choose what delivery method for what type of disease and work on what you can do in the meantime.
Thank you so much, sir.
Okay, next one. And okay, go ahead.
Hello, my name is Pim. I'm from Thailand, and I have a question about CAR T-cell therapy. So current CAR T-cell therapy can be used to treat cancers like leukemia and lymphomas. In the future, what are the opportunities and approaches to use CAR T-cell therapy to treat large tumorous and solid cancers?
Yeah, so that's been the biggest interest of every CAR T lab in the world, which is how do you get them to target solid tumors. There are many difficulties with it. One of them is, so when you're treating leukemia, the leukemia is in the lymphoid system, it's in every tissue in the body. So there's plenty of antigen to activate the CAR Ts. For a solid tumor, the antigen is just in the tumor, and the tumor has an immunosuppressive microenvironment. So we have to alter the CAR Ts to make them go into the tumor and to survive the immunosuppressive environment. So we're adding a variety of cytokines, some people call it armoring, to make the CAR Ts work better. We have vaccines that activate the CAR Ts against the tumor antigens. there's just going to be a lot more research needed to be able to treat solid tumors.
All right, thank you so much.
Okay, next one gonna be the last question. I apologize for the other student. Okay, next one.
Hi, Professor Weissman. Thank you for your insightful sharing. I'm Yena from Singapore, and you mentioned earlier about in vivo gene therapy for genetic diseases, and I was wondering whether you ever tried in vivo gene therapy for prenatal cases, and whether treatment of genetic diseases is better the earlier that it's been conducted.
No, you're absolutely right. And we've looked at treating fetuses in the mother two ways. One is you can you can cannulate the umbilical artery that delivers blood to the placenta, which crosses over to the fetus. We've also are working on formulations of LNPs that selectively target the placenta and cross into the fetus. So both of those can be easily done. You can also do larger surgeries and directly inject the LNPs either into the baby's blood or liver or other organs. And all of those are being investigated.
Okay.
All right, thank you.
Okay, thank you.